So yeah, and for the opportunity to talk to you all today. So Gil gave a really good introduction to the principles and the technology of how it works, and a really solid use case. So I suppose all I'm going to do here today is talk about how we've used the commercial version of this instrument, what we think it's good for, and just show you some nice images as you're all wilting in the 40-degree heat. Yeah, as introduced, I'm Alex Corbett. I'm a Royal Society Industry Fellow, full-time academic at the University of Exeter, but I'm giving this presentation on behalf of Cairn Research, who have created the commercial version of this technology. So just a bit of the brief history, just to recap as to how this came about. So we have the timeline there for Gil's developments that led towards the first publication of the RAP concept in eLife in 2021. We then sort of started tinkering around with our own version here in Exeter, and just to make the distinction easier, we gave it a name, the Multiscope. So, and that was the original Exeter Multiscope. So if you're looking through the literature, that's the reason for the distinction. It's the same technology. We then got the interest of Cairn Research, signed an evaluation license in 2024 and a full license in March 2025. And then just 12 months later, we had the official launch of the Multiscope, which you can see there on the right-hand side. And the working principle, as Gil's already covered there, just hide this, is that basically we can switch between different well positions indicated by the green squares there, just by switching lights on and off. Okay, and so we can use the same camera to be able to sample each well at the full camera resolution. Again, what do we think the unique advantages are? There are lots of high throughput, high content images on the market. What's special about the Multiscope? Well, it's not. We thought the initial advantage would just be raw speed, as we don't have to move anything mechanically. Since starting this project and talking to end users, one of the key advantages is actually the fact that the sample doesn't move. And we found that to be, again and again, talking to people, one of the key factors that makes it interesting to them. So the sample doesn't move. It's a fully controlled sample environment, so we can have it with humidity, temperature, CO2, nitrogen control as well. It's either sort of a phase contrast or wide field fluorescence imaging modes. So it's very gentle on the sample. That means you can do long-term studies. It does have the speed component there, so it can acquire data up to 100 times faster than any other system. Really the only bottleneck is the frame rate of your camera. And so if you've got a 100 hertz camera, which most scientific cameras can go to these days, then you can get a whole 96 well plate in less than a second. But how you use that bandwidth is entirely up to you. So whether you want to look at your sample in different colors, or if you want to use that bandwidth to acquire Z stacks across the plate, then obviously that you have that flexibility. It's been intentionally designed with robotic loading and unloading, so if you want to move between plates and to integrate with whatever your current workflow is. The third unique advantage we think compared to other systems out there is it's very flexible. So we can change the array of objective lenses from 1x to 20x. We can image anything from cells to whole animals. As you can see at the bottom there, we've got the zebrafish in a single well of a 96 well plate. It doesn't need any special environment, doesn't need to be in a dark room or on an anti-vibration table. And yeah, so it means you can, if you want to swap out a camera, one that has a large chip area to one that goes very quickly, then obviously you have the flexibility to do that as well. We've been using these systems for a while now, and so we've had the opportunity to work with a number of different research groups to get data to demonstrate its usefulness. So this means that particularly when that fact that the sample doesn't move and it's very gentle is particularly useful for cell suspensions, where you don't really want to accelerate your cells around and move them mechanically to the objective. Organoids, which are very tricky to grow, or can be, and this example below here is just taking a Z-stack through a cluster of different organoids, growth assays and co-cultures, etc. We can do fluorescence imaging. We've got images there of a fluorescent pancreas in a zebrafish. Hopefully you can see that, as well as some fluorescent HEC cells. We can do label-free imaging as well using this phase contrast modality. And as Gil pointed out brilliantly, we can also look at, if you want to look at dynamic changes within a particular sample, then it's obviously very well geared up for that, whether that's tracking applications, cardiomyocyte monolayer contraction, as Gil showed, or any optogenetic stimulation. This is the design that Cairn have come up with. They've made several modifications to the original implementation. The key changes that they've changed was to triple the focal length of the parabolic mirror, which produces significant improvements in the image quality, as well as having a second relay mirror, which is the big round elliptical one that you can see there. And that means it just gives us a lot more flexibility in where we place the camera in the system by having that fold. The system that we have at Exeter has a photometrics kinetics camera with a nice big 21mm square chip, and that can go up to 500 frames per second. So if we want to do tracking across 24 wells at 18 hertz, we can do that. Okay. And it also includes the incubator for long-term studies. So the primary applications at the moment, the systems that are commercially available are this phase contrast imaging modality with fluorescence available towards the end of this year. I can show you some fluorescence images later on as well from sort of a development system that I've got going in my lab. So what's the magic sauce that gives us this nice high contrast in our images? So we can see here on the left, we've got the same samples left and right. These are little annelid worms, sea worm larvae, and the only difference between these two systems is that on the right we've introduced a diffuser, which is lower down here, to increase the range of angles of illumination of the sample. And we can see that reduces the contrast in the image quite significantly. And I'm more than happy to go into that. We can go into that in the questions if we want to, as to why that is. We've got some slides at the end we can discuss. But basically, we by providing a very strong collimation of our LED source by using these collimation lenses, we are then very sensitive to any phase variations across the sample. And so wherever there's a phase gradient, we will see that interpreted into the intensity in the collected image. Okay. So we've got some cells. Obviously, we're very interested in imaging cells as well. Here are just some 10x images of a senescent cell culture. Each of these is 1.4 by 1.4 millimeters for a single well. And this is just a through focus stack. Each of the multiscopes comes with its own Z range of about 15 millimeters with 400 nanometer step size. So we can get these nice smooth Z stacks. And of course, we're imaging them in quick succession. So as well as just one, we don't have to just get one. We can get 2 or 4. You kind of get the idea, or 8, and anything up to 96. So this is a 96 well imaging system, and we can because it's so quick to switch between wells by switching LEDs on and off. It means that we would acquire each depth, acquire the full 96 well images, move to the next depth, and then reacquire. So we can get the Z stacks across 96 wells very quickly. For a single depth, then you'd have something of the order of 24,500 by 16,384 pixels, effective sort of composite image of your sample across the well plate, which gives you a space bandwidth product of around about 1.2 times 10 to the 7, which is about 2 orders of magnitude larger than you'd get in a single confocal image. So there's a lot of data there, is all I'm saying. It's because each well is sampled at the full camera resolution. Okay. And we can look in more details. This is 20x now. So here we're able to just taking a little shallow Z stack through these fixed cells. You can see the nuclei here. We can see some detail within the nucleus as well. And so we can use that for, if we want to get, use this small Z range to extract phase contrast images as well, using transport intensity equations that's been done before in the past. So we could extract quantitative phase from these images should we want to. This is also showing that as well as small, we can do tracking across small objects. These are algae, and also shows the sort of the relatively shallow depth of field that we can get at higher magnifications. So we're just only seeing the algae within the focal plane here. As mentioned before, we can look at organoid imaging, and this is very useful for high throughput where we want to look at the development over time. And so we can just image across multiple wells, and we can identify which ones have developed successfully and which ones haven't. I couldn't tell you which ones look good from that image, but we'll leave that to the experts. As I showed before, we can also get a Z stack, and this is important in this case because sometimes you get strange situations like this, whereas instead of just one organoid, we can have 2, or in fact we have 3 slightly different sizes sitting on top of each other, and that means we can get a sort of a rough depth coding of each pixel in the image to help interpretation. Another flexibility that we have with the multiscope, if we go back to the original design, is that the magnification that we see on the camera is just the focal length of, well, what is effectively the tube lens, which is the focal length of the parabolic mirror, to the focal length of the objective. So these objectives are placed one focal length away from the sample, and they don't necessarily need to be the same for each well. So you could have a well-to-well variation in magnification, should you choose. And just to illustrate that, we have here 3 different zebrafish imaged at 3 different magnifications. This is for a cardiac imaging study with Matt Winter and Siobhan Monaghan at the University of Exeter. And here we're just using a small region of interest around the heart of the zebrafish to extract the beat rate. We can do that at several different magnifications, so 3x as well. We can process that data, extract a beat rate as well, so quite robustly and reliably, just using the changes in the pixel intensities. And again, this talks to the use of phase. So we're very sensitive to any phase changes along the optical path. So anything we don't need to focus on the heart of the zebrafish to see these changes, and we can be out of the focal plane, and we could still see the beats within the animal. And we can go right out to 1x magnification. So if we want to see the entire well of a well plate and ensure that we always have the animal within our field of view, then we can do that. And remarkably, we can actually still extract the beat rate from this from these images because we're sampling at the full camera resolution. Some more recent data that we've had is, again, looking at these nematode worms, very popular animal model. And we can look at this at 10x magnification. We get this, because of this this phase contrast effect, we get this lovely contrast of the animal against the background. You see a little bubble grouping in there. But that allows us to extract lots of information very easily in the post-processing in terms of the pose of the animal, its its direction of motion, or for other animal models, its field of view as well. Okay. Again, going back to these annelid worms, we can see here, we can look at these at higher magnification, and this, even though we've got just 4 fields of view here, we're actually sampling 16 of these at 450 hertz, which gives us a very high frame rate per well, which means that we were able to track them with high accuracy, even when they're moving quite quickly. And again, we can go back to this 1x imaging mode if we want to make sure that we never miss a single animal and constantly track it. These other bits in here are the food, by the way, just in case you're wondering what they are. So this is what's making this animal quite so pleased with itself. Okay, so it's having a good lunch. So I'd mentioned we'd look at fluorescence imaging. So here on the left, we've got another higher magnification view of these annelid worms. On the right, we have a shallower Z stack through one of them that's expressing this GFP in its nervous system. And we sort of combine these 2 views here. So this is, again, wide field fluorescence imaging. So it's, we're not seeing distinct optical sections, but we can get by taking a Z stack, we can sort of get a feel for its distribution across the animal. Okay, we can do cells as well. So this is looking at cellular level fluorescence. So beads are easy. It's harder to image fluorescence in cells. Here we've got a study with Ben Houston and Nisha Era, and we're looking at this variable expression of this GFP reporter across these cells. We can sort of get this is the phase contrast version of a similar field of view, and just see cell-to-cell variations in this fluorescence brightness. We're also pushing this to see what the limits of sensitivity are. I know we've had a question similar to that before. What's the smallest feature we can see? What's the smallest amount of fluorescence that we can detect? And one of the projects that's really helped us to push the envelope on this was one we had last year with a company called Cytocom, who were looking to measure cell health in bacteria using an assay whereby they have these electrodes that sit very close to these bacterial colonies. They provide an electrical stimulation through the electrodes, and if the cells are alive and healthy, we see an increase in this fluorescent signal, and if they are not, then we see this decrease. And so the question was whether we could have the level of sensitivity to extract that. And so we built a custom system. This is not the commercial multiscope. This is just to indicate where the limits are. So we didn't have the relay optics, and you can see why the relay optics are a good idea, because when you've got a big chunky fluorescence camera in there, it could start to obscure the optical path. So here we're using UV LEDs with an excitation filter, and we've been just collecting using shorter focal length, sort of compound objective lenses, still on 9mm diameter, so we could fit within the 96 well pitch, and using some fairly strong excitation and emission filters, we were able to extract out the difference in between alive and dead cells. And the whole purpose of this project is that having this electrical stimulation assay, we want to be able to parallelize that across 96 wells rather than just do one at a time. There's a link to the paper here. So again, we can, whilst we were, this shows the individual bacterial colonies. We weren't resolving individual bacteria here, but we could certainly see how the individual colonies were changing over time. Okay, so a couple of minutes left. So that's just like I said, thank you to all of the collaborators that we've worked with. I think, as I said, a lot of the magic we're getting from this is from the LEDs, which offer a variety of different source wavelengths and very small emitter sizes that enable us to image samples with such high contrast, as well as using them for fluorescence imaging, even in the UV, as we've just seen, providing the spatial coherence to allow us to see even very weak phase objects, being able to exploit the developments of new cameras to acquire images at very high frame rates, and fluorescence imaging we can acquire as well at very high frequencies. Obviously, it requires slightly longer integration time for biofluorescence than you would for just a bright field imaging. So we have actually this is now out of date. We have integrated stage-top incubators for long-term experiments, and we are now trialing the Multiscope several sites. We've had one in Exeter since March this year. There'll be one available in Glasgow at the bioimaging workshop there in late July. One in Cambridge from August this year, and we're looking for other UK sites as well. So if you're interested in using the Multiscope at any of those locations, then you can follow the QR code to make a booking. But this is me with the Cairn Research team. So just right at the start of the project, when we're just working on the very beginnings. But thank you very much for your time and for your attention. I believe I've got 10 minutes for questions now. Thank you.